Hello everyone, welcome to the ITLS Academy. Today we are going to discuss the topic that is your aerobic microbial count. Okay, microbial count in a water is also known as a aerobic colony count or total viable count or aerobic plate count. Aerobic plate count. It is the another name of the aerobic microbial count. Aerobic colony count, total viable count, or aerobic plate count. It is simply a microbiological testing, which is used to measure the which is used to measure the number of cultured microorganism present into a sample. It can also use to detect the total number of total number of bacteria, yeast, or mold inside the water present, not inside, present into the water. Just a minute. Present into the water. This aerobic microbial count also used to decide the quality of the sample. Okay, if the microbial load is too high, then obviously the quality of the product is bad. Okay, and if the microbial load is not too high, then in that case, the quality of the product is good. So that's why it is very important that to determine the aerobic microbial count. And this aerobic microbial count are detected by the culture media. This culture media is what? It is the food for the microorganism, or you can say that the substance which is used for the growth of the microorganism. Okay, the substance which is used for the growth of the microorganism. And this microbial count are also under come the category of the microbial analysis. Okay, when we do a microbial analysis, ultimately we decide the quality of the product. So let's start the lecture. Principle, first of all. So plate count agar is general purpose growth medium commonly used to assess the total or viable bacterial growth of a water sample. So simply this uh, plate count agar is what? It is a general purpose growth media. Okay, the common type of the growth media is what? It is a plate count agar media allow the which allow the growth of the all type of the microorganism. This plate count agar is used as a general purpose for the growth of the media, and it is simply or commonly used to access the total or the viable bacterial growth present into a sample. The number of microorganism per millimeter of the sample is calculated from the number of the colonies obtained. Okay, so in a plate, how much colony we obtain is used to determine the number of the microorganism per millimeter present into a sample. Okay, and it will be done by a serial dilution. Serial dilution. So With the help of serial dilution, we choose the dilution. Okay, suppose that 10 to the power minus 1, minus 2, minus 3, minus 4, minus 5, and minus 6. In general, we do a serial dilution from 10 to the power minus 1 to 10 to the power minus 6. Okay, so with the help of the selected dilution, we calculate it. Now, pour plate are prepared using a specified culture medium and a specified quantity of the sample. The plates are aerobically incubated at two different temperature, that is 37 degrees Celsius for 24 hours and 20 to 22 degrees Celsius for 72 hours. Okay, so we put the plate inside the incubator at two different temperature. First 24 hour we set the temperature 37 degrees Celsius, and after 24 to 72 hours we change the temperature between 20 to 22 centimeter and allow the growth. Now the next is your culture media. So here we required a plate count agar media, buffer peptone water, and overlay media, that is your agar media. Now what is the composition of the composition of the plate count agar? Okay, so we have to know it. Basically, plate count agar contain yeast, glucose, sorry, yeast, glucose, agar, distilled water. Okay, so if the distilled water is around 1 liter, then agar, amount of agar is 15 gram, glucose is 1 gram, yeast extract is 2.5 gram, and one more thing, that is tryptone. Tryptone is around 5 gram. So this composition is for a 1 liter. Okay, this is the composition of the plate count agar media. Buffer peptone water we have to use, and agar medium will be used. Now what is the composition of that agar medium? So basically the composition of that agar medium, for 1 liter we have a peptone, beef extract, sodium chloride, agar, and distilled water. So if distilled water is 1 liter, then the amount of peptone is 10 gram, beef extract also 10 gram, sodium chloride 8 gram, and agar 15 gram, and distilled water is 1 liter. So this is the composition of the agar medium. Okay, so once the media is prepared, then we have to adjust the pH is around 7.0. Same, its pH will be 7.2. Then after that we autoclave the media. Okay, so why we autoclave the media? So that if any contamination are there, then it will be eliminate. For eliminating the contamination, for eliminating the microorganism, we autoclave it. Okay, so if you don't have a plate count agar in your laboratory and you have this ingredient present in your lab, then you will prepare the plate count agar. Same with agar media. If in your laboratory there is no proper box of the agar medium are there, then with the help of these ingredients you can prepare it. Okay, so this is about the culture media. Now the next is your procedure. Disinfect the surface of the bottle, pouch, cup containing sample with the 70% of the ethanol thoroughly. Now mix the sample by a vigorous shaking to achieve a uniform distribution. Now what you have to do, you have to clean the laminar airflow. Okay, how we clean it, the whole video are already present in our account. You can go and check it. Okay, now you have to sterilize the media inside the autoclave, and you have to sterile the glassware inside the hot air oven. And how you do it, the video related to this topic are already uploaded. Then after that, the whole experiment will be performed inside the laminar air flow. Okay, now what you have to do, you have to aseptically inoculate the 1 ml of the sample using a sterile pipette into a sterile Petri plate in a duplicate in 2 sets. Now, Petri plates should be labeled with the sample date and any other desired information. Okay, if you do a pour plate method, which is mostly we perform, so in that case, what we have to do, we have a sterilized Petri plate that is sterilized inside the hot air oven. And how we sterilize it, you can check our video. Okay, after sterilization, what you have to do, you have to take a sterile pipette and pour 1 ml sample inside the Petri plate. Okay, after pouring it, you have to pour the media into the Petri plate. This is our pour plate method. Now you have to mention the sample number, date, and other information like your name, your batch. If you want to mention it, then you can mention. Now pour into each plate around 15 to 18 ml of the molten sterilized PCA media that is cool at 44 degrees Celsius to 47 degrees Celsius. That I already told you what you have to do after pouring the sample. You have to pour your PCA media. Now avoid the pouring of the molten medium directly onto the inoculum. Inoculum means that the sample that we are putting it. Now mix the media and inoculum by swirling gently clockwise and anti-clockwise so as to obtain a homogeneous distribution of the inoculum in the media. Now we put the Petri plate inside the laminar airflow so that it will be solidify. In case wherein sample microorganism have spreading colonies is expected, then add 4 ml of overlay medium onto the surface of solidify plate. After complete solidification, you have to put the Petri plate inside the incubator at 37 degree Celsius for 24 hours, and the Petri plate should be placed invertly. Okay, then after that, after 24 hours, you have to set the temperature 20 to 22 degrees Celsius for 72 hours. After specifies incubation period, count all the colonies including up pinpoint colonies. Now when you get the when you get a after 72 hours, what you have to do, this is your Petri plate. Now the colonies grows like this. Now you have to count all the colonies, even pinpoint colonies. Now spreading colonies shall be considered as a single colonies. If less than 1/4 of dish is overgrown by spreading, count the colonies on the unaffected part of the dish and calculate the corresponding number in the entire dish. If more than 1/4 is overgrown by spreading colonies, now discard that plate. Now calculation, plate having a microbial count between 10 to 300 CFU. Now in that case, n is equal to colonies plate 1, colonies plate 2 divided by 2. Plate having microbial count less than 10 CFU but at least 4. Calculate the result given in case number 1, same as it is. If the microbial load is from 3 to 1, then reporting result will be a microorganism are present but less than 4 per ml. And if the sample plate contain no colonies, then reporting of the result shall be less than 1 CFU per ml. This is how we calculate the colonies. So this is all about the aerobic microbial count, how we detect it, what is the principle behind it, what is the procedure. I hope you will understand, but if you have any query related to this topic, you can ask me in a comment box, or for a further detail, you can follow the ITLS Academy on different platforms, that is Instagram, Facebook, Twitter, YouTube, WhatsApp and LinkedIn, and subscribe our channel ITLS Academy on YouTube and get our app from a Google Play, or you can subscribe our, you can also ask your query to the helpline number that are already mentioned in our slide, that is 7080833450, and visit our website www.itlsacademy.com. Thank you for watching. Please like, share and subscribe our channel.